Okay, so hello everyone. Welcome to today's ImmuZoom seminar. Again, I want to really appreciate today's speaker, Jack, to spend the time for the weekend with us and then share his science with us, and also obviously you guys to be with us on the weekend. It's really my pleasure to welcome today's speaker, Dr. Jack Bill from Department of Pathology in UCSD. So Jack currently is a professor, as I just mentioned, in Department of Pathology in UCSD. So Jack got his bachelor degree in UC Berkeley, and then continue to pursue his PhD and MD study in both in UCSD. So from 2002, I think Jack started to start his postdoc training in WashU, St. Louis. And at the same time, he also started his clinical resident training in the clinical chemistry and the clinical immunology, also in St. Louis, in Barry's Jewish Hospital. So from 2006, Jack got his first position and started his independent research program as assistant professor in Department of Pathology in UCSD. To this day, he's a full professor full professor in Department of Pathology in UCSD. Jack's lab mainly study how to initiate innate immune responses to cancer. His basic research lab used mouse and human model systems to elucidate genes involved in tumor rejection. They focus on innate immune cells and also signaling pathways in the cancer cell itself that can mediate anti-tumor immunity. And then their project encompasses myeloid cell biology, innate lymphocytes, tumor-derived cytokines, and cell lineage studies. I think Jack's lab actually published a series of different papers elucidate the tumor-expressed cytokine IL-17D in promoting tumor immunity, and their study continued to elucidate that pathway upstream and downstream of IL-17D, and also extend its role into viral and then bacterial infections, allergy, and autoimmunity, which I think we're gonna hear a lot about today. They also focus on the role of interference in tumor growth and rejection, establish a linear tracing model to study the role of interferon signaling in tumor progression. More than that, as I just mentioned, Jack actually also get extensive MD training. So he currently also directs clinical flow cytometry, diagnostic immunology, and stem cell processing core. He's clinical interested, including bone marrow transplantation, stem cell regeneration, and autoimmune diseases. They have examined longitudinal samples from different patients undergoing different immune therapies, and have established a pipeline to measure activation of innate and adaptive immune cells from human blood by using flow cytometry. So I think Jack actually did a series of wonderful work on both basic and clinical research. Based on these, he got numerous awards and then honors, which including continuously he got nominated as a UCSD School of Medicine Kaiser's Excellence in Teaching Awards. And then he also got AI laboratory travel awards for both 2017 and 2018. So without further ado, Jack, thank you so much be with us today, and then really exciting to your seminar. Thank you so much.
All right. Thanks, Sean. That was a really great and thorough introduction. I obviously won't be able to talk about all the different aspects of the research. I'm focusing really mostly on a couple of different stories. I want to get you up to speed with the IL-17D story, which is published, and then the extracellular vesicle story is not published at all. And we're very excited, wanted to get your comments on this. Since this is kind of a weekend seminar, I want to kind of be a little bit informal, and if you have any questions, go ahead and shout them out. I'm happy to stop and take any questions throughout the talk. So I wanted to start out with just a couple of case studies to set the stage for what model system my lab uses and why we chose this model system. This is a really old case study published in 1940s, and it's about spontaneous regression. What you see is this 5-month-old baby girl who has this mass here that's been growing steadily since she was 2 months old. And so she was taken to the hospital and admitted, and largely everything was normal, no fever, the labs were normal. All they found was the tumor. And so already you kind of know that this is not a good diagnosis, because at that age, there's not many things that can cause this growing mass. And so malignancy is at the top of the list. It could be infectious. In any case, they did a biopsy. And the idea was that if the tumor was benign, then they could just remove the tumor. But what they found was that it was actually invading muscle, blood vessels, and the nerve. And when they sent the biopsy for pathology, it was diagnosed as fibrosarcoma. So there's not much you can do for that. The wound was closed, and it was decided that they wouldn't do anything. The parents were given this hopeless prognosis. But what eventually happened was that the wound really healed, and the tumor, rather than growing larger, ended up regressing, such that at 12 years of age, you can see the X-ray here, there is really no tumor at all. Okay, so this is a case of bona fide spontaneous regression of a tumor. Okay. So that's kind of one case. I just want to mention that. The second case, I chose this just for the picture here. This is a clinical trial with a dendritic cell vaccine. I just chose this mostly to illustrate two things. First is that clinical trials with immunotherapies have been largely small, especially novel immunotherapies. This trial had 21 patients. And the response is rare. These two arrows indicate the 2 patients who responded. And you can't learn much, actually, from these 2 patients because it's small and you don't know why these patients responded. Okay. But the point of this trial was that in these 2 patients, the cure is miraculous. You can see this patient was diagnosed in the year 2000, had metastatic melanoma, underwent surgery, interferon therapies, several different clinical trials, even had a peptide vaccine before enrolling in the current vaccine trial, and then just had an amazing response. Okay. And so those 2 case studies illustrate what I call successful anti-tumor immune responses. Okay. In the one case, it was successful, but we didn't really do anything. This is called spontaneous regression. The tumor regresses without treatment on this documented cancer. And this is, of course, very rare. It's thought to involve immune cells and potentially other mechanisms. And then we all know about cancer immune therapy, right? This is tumor regression after treatment. At least for immune checkpoint inhibitors, it's, the success rate is lower. It's low, it's 20 to 30%. There's a third type of successful anti-tumor immune response, and that's what my lab initially started studying. And this is called tumor surveillance. And tumor surveillance is defined as tumor regression without treatment, just like spontaneous regression, but it's clinically silent. Rather than having a documented cancer, tumor surveillance occurs in healthy people, right? It could be happening as we speak to us, as we're here sitting, listening, and being healthy. We don't really know the incidence. How often is it happening in us? Are we constantly getting rid of cancer cells in our body and not knowing it? But obviously, we think that it involves adaptive and innate immunity. And so I like to study this tumor surveillance process in part because I hope that it'll kind of begin to define how we can make cancer immune therapy better. So I'd like to study tumor surveillance. I want to first just start out historically, what is the evidence that this even exists if it's clinically silent? How do we observe or even model a clinically silent process? And so I'll just go over just a couple of historical data on the fact that tumor surveillance does exist. Okay? And there's 3 main lines of data. One is that if you take out the immune system, you get more cancer. The second is if you look at people with cancer, you can see tumor-specific T cells inside the tumor. And these T cells actually are doing something because they're associated with a better prognosis. Okay, and so I'll just give you, a table showing the first piece of data here, and this is looking at transplant patients. So these are patients who received allogeneic organ transplant, solid organ transplant. They have to be immune suppressed for the rest of their life to prevent the immune system from rejecting the organ. And these individuals get more cancer. You can see that, in this cohort, non-melanoma skin cancer, they observed 127 cases compared to 5 cases that, would be of a control or historical cohort. And so there's a 25-fold increase in cancer in all of these cancers here in these patients, 25, 14, et cetera, et cetera. Okay. So just like opportunistic infection, which is more in immune suppressed individuals, cancers are also opportunistic. This is a mouse model. This is from Bob Schreiber's landmark paper showing that immune deficient RAG2 mice get more cancer than wild type control animals. Okay, these are genetically identical, except this one has no immune system. And these other immune deficiencies also lead to more cancer. Okay, so this is a classic model where we inject this carcinogen and we look for tumor formation. And the immune system is actively preventing tumor formation. In fact, wild type mice with an immune system, I would argue, are fairly resistant to carcinogen. Only 20% of them actually develop the cancer. You don't have to use a carcinogen model. You can just let the mice sit in the animal facility. And if you let them sit for close to their lifetime, close to 2 years of age, you can see that the wild type animals have no tumors, or some of them have adenomas. But every single immune deficient animal has some form of tumor, adenoma or adenocarcinoma. And then this RAG stat animal further is immune deficient, and it gets lots of adenocarcinoma. So those really landmark studies showed that cancer immune surveillance exists. And why is that important? It's because when we look at people who have cancer, then there must have been a failure of cancer immune surveillance. Okay. And arguably, this failure of cancer immune surveillance is the first step of cancer immunoediting. And so I'll introduce cancer immunoediting. I did my postdoc with Bob Schreiber, and in his lab came up with the term cancer immunoediting. And we published this snapshot in Cell just recently to summarize the cancer immunoediting steps in the context now of immunotherapy. And so cancer immunoediting starts out here in the top left with cancer cells that some of which are recognized by the immune system. And when they're fully recognized and destroyed, they could be eliminated. And this is tumor surveillance. This is done. That means that, there's no more tumor. But what happens, we think, in people who get cancer is that this elimination process is incomplete. And so some of these tumor cells survive and they undergo this equilibrium process, which could last for years. And we have this model in the mouse as well, and it could last for several months in the mouse. And eventually, these cancer cells then escape the immune system and they grow out. And this is what we see clinically. Okay, so by the time we've seen a patient, right, presumably these steps have already happened in that patient. They've had an unsuccessful tumor surveillance process. And then these patients we can treat with checkpoint therapy or other types of therapy, and we can get a complete response. That's the second elimination phase. But more often we have non-responders and we have acquired resistance, and these are some of the characteristics of these cancer cells. Okay. And so the question I was interested in since I was a postdoc was, what about these cells here that were destroyed in elimination? What are the characteristics of these cancer cells here that make them immunogenic? And the problem, of course, is these cells are dead, right? I wish to study, what are their characteristics? And so what is the model system that we can use? And so here's our model system. This is injecting animals with methylcholanthrin, and you can do a dose response. And you can look at wild type or RAG by gamma chain knockout. And so I was interested in the innate immune system, so I introduced a further immune deficiency here in this RAG by common gamma chain knockout. And you can see that the wild type animals, again, are fairly resistant to cancer. This is % tumor free. And then the RAG animals get a little bit more cancer, and then the RAG common gamma chain have a little bit more cancer. Okay, and whatever dose you use, you get kind of the same results. So what does that mean? how do we take advantage of that? So here you're giving MCA to wild type animals. And here I'm showing, maybe half of them get cancer. And here you're giving MCA to immune deficient animals and more of them get cancer. Where is tumor surveillance occurring in this model system? Which animals? It's actually these animals over here, right? Tumor surveillance is a silent process and it keeps you healthy. And so these wild type animals have already gotten rid of their tumors. But those are the tumors I exactly want to study, but they're gone because they've undergone tumor surveillance. And so what you postulate is that tumor surveillance is absent over here. So some of the tumors that form here are really highly immunogenic, right? They would have been destroyed if there was an immune system, but they weren't, okay? And so as a postdoc, I generated about 50 cell lines from these immune deficient animals. So each of these tumors you can harvest, you can generate cell lines, okay? You can generate from wild type animals. And then you can put these cell lines back into another animal. And I postulated that there were immunogenic cell lines that would have been destroyed and they wouldn't ever form a tumor. Whereas those that escaped the immune system would be able to form a tumor, okay? And that's exactly what we found, was that we were able to identify regressor cell lines, regressor tumors from these immune deficient animals, that when you transplant them into animals, they underwent spontaneous rejection by the immune system. Okay, and then progressor cell lines were similarly developed, but in a wild type animal. Okay, so that was our model system. We had regressor cell lines and progressor cell lines, and over the course of several years, we harvested 9 different cell lines from wild type hosts, about 10 different ones from RAG hosts, and another 10 from RAG gamma chain hosts. And then we transplanted these cell lines back into a wild type animal. And every single cell line from a wild type animal could still grow. They're all progressors. Some of the cell lines from a RAG animal, 3 of them were regressors here, shown in the open symbols. They are rejected in wild type hosts. They grow fine in RAG hosts. And even more were regressors when we took out them out of these RAG gamma chain mice. Okay. And then when we do kind of a cumulative experiment over, I think this is like about 4 or 5 years, looking at 9 cell lines to 87 mice, these cell lines all grew very well when they're derived from wild type animals. But when you generate cell lines from immune deficient animals, you can see 15 cell lines we tested here in 120 mice. You can see that some of them fail to form tumors. And then from the RAG gamma knockout animals, even had more of them fail to form tumors in a wild type host. In a RAG host, they all form tumors. Okay, so these regressors are undergoing spontaneous rejection by the immune system. Okay, and so what we think happened is that these should have been destroyed, but they're not, and we were able to rescue them. And so we think that these are actually models of these original nascent tumor cells here that are destroyed by immune recognition in a wild type animal. But in a RAG animal that lacks these immune components, these immunogenic cells can grow out and you can isolate them. Okay, so that's our model system. And so I've been working on this for the last 15 years or so. And of course, we want to know what are the molecular differences. And there's lots of papers published, especially from Bob Schreiber's lab, on how regressors have different antigens they can bind to and be presented by MHC class 1, and the antigens can be edited. Checkpoint blockade can also edit these antigens and reveal more antigens. I want to talk about 2 stories. One is that regressors have more adjuvants. This is published, so I'll go through it quickly. And then the other one is that regressors secrete these special extracellular vesicles. So how do I show that they have more adjuvants? We did this study where we took a progressor and it grows fine, and we mixed it with a regressor. Okay, so progressor D30M1 is mixed with these 2 different regressor cell lines here, D30M2 or D42M1, and in both cases, the mixture doesn't grow as well. Okay. This mixture requires it to be in the same spot, because if we immunize first with the regressor, then we come back with a progressor, you can see that it grows fine. And we know that the cell lines are not antigenically related. Okay. And so it has to be in the same spot and the same timing for this to work. And so when we did a microarray study, we found that IL-17D was expressed in regressors, but not in these 16 progressor cell lines that we looked at. In the 8 regressor cell lines, it was highly expressed in most of them. So that's how I started working on IL-17D. You all may know that the IL-17 family has lots of different activities. IL-17A and F are the ones that are most well studied. They're made by Th17 cells, and they act in terms of antibacterial and antifungal immunity. IL-17E, also known as IL-25, fights parasites. But IL-17D still is not very well studied. We know the receptor now, one receptor is CD93. And at that time, when I started studying this, we knew that it was expressed outside of the immune system. And so it was not, I guess, out of the concept that tumor cells could express 17D, especially regressor tumor cells. And so we overexpress IL-17D. So here's a progressor here. It grows fine. If we overexpress it, you see it has problems growing. We have 4 different cell lines here, and to some extent, it can delay tumor growth. Since this is published, I'll just show just a couple of slides and show you a summary. How it works is that it recruits NK cells. Every time you overexpress it here in these different cell lines, you get more IL-17D. And then when you inhibit it, then you get more NK cells. And when you inhibit 17D, you have less NK cells. And so mechanistically, we showed that IL-17D made by a regressor cell acts on endothelial cells to induce CCL2 or MCP1. And this recruits both monocytes and NK cells and probably T cells as well. And we see more NK cells, we see more M1 type macrophages through interferon gamma. If you block interferon gamma, you deplete NK cells, and you block CCL2, then you can block IL-17D mediated rejection. Okay. And so if you mix this with a progressor, you can create this adjuvant effect where the local microenvironment is changed, and you have more M1 type immunity, and you can activate T cells now to kill both regressor and progressor cells. Okay. So that's how we got interested in IL-17D. We next wanted to study how is it that it's regulated? What makes a regressor produce IL-17D? Okay. And so this is also published, or this is led by Robert and Ruth in the lab. They've since left the lab. But what they found was that there are these NRF2 binding sites in the IL-17D gene, in both mouse and human, and it's conserved, and there's like 6 or more of these binding sites in both mouse and human loci. So what is NRF2? NRF2 is a transcription factor that's held in the nucleus by KEAP. KEAP has these cysteines that are hyperactivatable by reactive oxygen species or oxidative stress. And when you get these oxidative stress or these oxidants, then you can cross-link these cysteines, and it releases NRF2 to go to the nucleus. Otherwise, NRF2 is brought to the proteasome and degraded. When you release NRF2, it gets to the nucleus, it stimulates these antioxidant response genes, these antioxidant response element containing genes. And so we know it's studied quite a bit. NRF2 is a primary regulator of the antioxidant response. It induces detoxification genes and other antioxidant genes. And there is some role in immunity, but what has been published is that it's largely kind of anti-inflammatory because it's thought to actually reduce free oxygen radicals, which are pro-inflammatory. And so could it regulate 17D, and is there physiology there? And so there are a couple of NRF2 agonists. Hydrogen peroxide is one. TBHQ is the other one. This is the structure. This is a nice electrophile, and so it can really stabilize and stimulate NRF2. And indeed, treating with hydrogen peroxide or treating with TBHQ, you can stimulate IL-17D. Compared to here, this is a benchmark gene, HMOX, that is known to be a target. And so in both cases, you can stimulate IL-17D in vitro with these compounds. In vivo, we can see a correlation. So it's associated with evidence in vivo and human and mouse tumors here. This is just looking at antioxidant response element genes, and you can see a loose correlation with IL-17D. So probably there's other regulators of these antioxidant genes as well as IL-17D. But you can see regressors have more 17D, as I showed, and they also have more NRF2. So can we activate NRF2 and stimulate tumor regression? We can do that with TBHQ. This is an experiment where we have bilateral tumors growing here, and we just paint TBHQ onto the tumor, or here we have the control. And so you can see that you can get some limited growth delay of B16 melanoma by activating NRF2 through TBHQ. This requires the immune system, because if you put in a rag common gamma chain host, you don't see this. Interestingly, I don't show the data, but a rag host also responds. And so the NK cells seem to be sufficient to limit the response to some extent. And you can see that TBHQ leads to more NK cells in the tumor. We were interested in which cells were responding. And so we did this kind of clever experiment, I thought, where we have B16 melanoma, which can activate NRF2 and IL-17D. We put that into a knockout host of NRF2 or an IL-17D knockout host. And in both cases, you still got a response. So TBHQ still works if the host lacks NRF2 or IL-17D. But now when you knock out NRF2 or IL-17D in the tumor cell and transplant to a wild-type host, now the wild-type animal should still respond to TBHQ. But in this case, TBHQ doesn't work as a therapy. Okay. And so we think that it acts specifically on the tumor cell, and the host doesn't really need to respond. And so this to us identifies kind of a tumor-mediated mechanism where the tumor could actually lead to its own rejection. We could find ways to induce NRF2 or IL-17D in tumor cells in a sustained way. We might be able to use it as an immune therapy. Okay, and so to this big model here, we're going to add NRF2 here. We still don't yet know why regressor cells have this pathway activated. It's possible they just have more oxidative stress than progressor cells. But now we kind of are thinking that this is how one way that tumor surveillance could occur is by activating this NRF2 IL-17D pathway. Okay, so now I'm going to go into some unpublished data. We're going to talk about this idea that regressors can also activate a systemic response. So we did this experiment where we had the mixture on one flank. And so the mixture undergoes rejection because the regressor is making IL-17D. Okay. If you actually blocked IL-17D in the regressor, you don't get rejection. I didn't show you that data, but this rejection requires IL-17D. Okay. When you have the progressor on the opposite flank, it still grows fine. And this to us said that this was co-localization, right? It needed to be on the same flank for the regressor to work. But in fact, since we've done these experiments, dozens, if not hundreds of times, we observed that this progressor actually grew a little bit more slowly. And we wanted to know, was this true that this progressor on the other flank was really growing more slowly? And so Steve and Preston and these other individuals, Calvin, Christine, and Jared, they all worked on this project. And we set up a system where we put a regressor on one flank, and then we put a progressor on the other flank. And we just looked at tumor growth. And in this first experiment, we had a mouse that didn't have a regressor at all, okay, as a control. And so the regressor, you can see here in a rag mouse, grows fine. And in a wild-type mouse, it undergoes rejection. So this is just looking at this flank for the regressor. On the opposite flank, though, you can see that here in a rag mouse, the progressor it grows fine. It doesn't know the regressor's on the other side. But in a wild-type mouse, somehow it knows the regressor's on the other side and it grows a little bit more slowly. Okay. If you put a progressor on the other side, it grows the same as PBS. I thought PBS was better because I didn't want there to be competition between progressors and the flanks and everything. Okay, but in any case, the mouse knows that there's a regressor, and the immune system responds accordingly and slows down the growth of the progressor on the opposite flank. Okay, and so that was the observation that led us to really think about exosomes or extracellular vesicles. So many of you probably know about these extracellular vesicles. This is kind of the background of that. They're derived from these multivesicular bodies, and so they're actually vesicles within a vesicle. Okay, we think that there's cargo that's loaded, and the cargo includes RNAs, proteins, lipids, just lots of bioactive molecules can be loaded into these exosomes. And then when these multivesicular bodies then fuse with the cell membrane, then you're actively exocytosing these small vesicles or exosomes. And it turns out that tumors make a lot more exosomes than normal cells. Okay? And so tumor-derived exosomes have been shown to do lots of different studies demonstrating pro-metastatic activity. For example, you can isolate exosomes from tumors, inject them into a mouse, and it can educate the bone marrow. And then when you challenge these mice with tumors, you can get increased metastasis if the exosomes came from a tumor that metastasized. And so the idea has been that tumor-derived exosomes have pro-tumor activities. And almost all of the previous studies have shown that these tumor-derived ECVs with pro-tumor activities, they've been done with progressor cells. So of course, the exosomes would kind of promote tumor growth. And so we had this question, we had this unique system of regressors. And so we were the first now to look at exosomes from regressor tumor cell lines. Okay. And so that's what we did. We had these 4 different tumor cell lines that were regressors. We had 4 that were progressors. And these are just cell lines grown in vitro. You can then isolate exosomes from these cell lines. And we did exosomes from 8 different cell lines. And we looked at the proteome, the proteome of these exosomes using mass spec. And so we found lots and lots of different candidates, but we focused on MEK1. MEK1, as many of you know, is MAP kinase. It's the only protein that was found in all 4 regressor exosomes and not found in any of the progressor exosomes. Okay. We decided to focus on it just because it was already known that it's a target for therapy. It's a tumor intrinsic oncogene. Okay, it promotes proliferation, and we even use MEK inhibitors to treat cancer. And so we thought it was counterintuitive that it would be in regressor exosomes, because if anything, it should promote cancer. And so we confirmed the proteomic studies with a Western blot. You can see that when you take host cell lysate, all of these cell lines have MEK1. But when you take exosomes, only really the regressor cell lines have MEK1 in the exosomes. The progressor exosomes don't have a lot of MEK1. And some of it is actually phosphorylated. Phospho-MEK1 is also present in these exosomes and regressors. And so we wanted to now test the role of MEK1 in exosomes. And so we decided to overexpress MEK1 to try to get into exosomes. And we knew that there were issues with this, because if you overexpress MEK1, potentially you would increase tumor growth. But we did it anyway, because we really wanted to have a system where we could have a progressor cell that was overexpressing MEK1, and that allowed it to express MEK1 in its exosomes. And so that's what we generated, was this progressor cell that had MEK1 overexpressed so that it extended to the exosome component. And we found indeed, and paradoxically, that these now MEK1 high cells grew more slowly. And they grew more slowly in wild type mice, but not in immune deficient NSG mice or RAG mice. And in fact, in this model now, when you inhibit MEK1, you actually get increased growth and not decreased growth in this MEK1 high tumor. Okay, and we know that some patients don't respond to MEK1 inhibitors, and you wonder whether these patients have regressor-like tumors that might actually have MEK1 in their exosomes. So we also isolated exosomes from these progressors. So now we have control exosomes from that are MEK1 low from the progressor, and then exosomes from the MEK1 high progressor. You can see that when you treat with this MEK1 high exosomes, you get diminished tumor growth. It happens in wild type animals, but not in rag animals. And you can see that the tumor is smaller when you treat the animals with MEK1 high exosomes. Okay, so MEK1 high exosome therapy can reduce tumor growth. It's interestingly associated with increased NK cells, slight increase in CD8 cells, and increase in M1 type macrophages, so that the polarization of the M1 type macrophages is much higher when you have MEK1 high versus MEK1 low tumors and exosomes. We wanted to see whether the exosomes were different. And so we isolated exosomes from MEK1 high and MEK1 low tumors. And you can see that they're identical in terms of their size, in terms of their morphology. There's no difference there. We even did a repeat proteomic analysis of these MEK1 high and MEK1 low exosomes, and most of the cargo is unchanged. 90% of the cargo is identical between MEK1 high and MEK1 low. But there are some genes that are higher in the MEK1 high exosomes, including MEK1, and then some genes that are lower. Okay, and so we can't say that the MEK1 high exosomes are acting through MEK1. We do know that if you inhibit MEK1 activity, that it blocks the activity of these MEK1 high exosomes. So how does it act? So we essentially treated splenocytes with exosomes, and we found that macrophages take them up. Okay, so that's part A. Part B, we can see that macrophages, bone marrow macrophages, actually internalize the exosomes. And we can transfer HA-tagged MEK into the cytoplasm of a macrophage by treating it with exosomes. So you're actually delivering MEK protein into the cytoplasm, okay, through these exosomes. And then when you look at a tumor-bearing mouse, if you injected labeled exosomes into it, you can see about 1% of the cells will take up the labeled exosomes. And these cells are all myeloid origin. They express CD45, CD11b, F480. And CD4, CD8s, and NK cells really don't take up these exosomes. So we know that we can overexpress MEK1 in tumors and force them to secrete MEK1 high exosomes. These tumors grow more slowly, associated with more NK cells and more like macrophages. We know that the exosomes themselves can be used as therapy. And if you block the activity, we think the activity is important. Then you block the activity of the exosomes. And we know that macrophages are taking up these exosomes. And so we then ask, what do the exosomes do to the macrophages? And so we did this RNA sequencing study. We treated tumor macrophages in vitro with MEK1 high exosomes versus MEK1 low exosomes. And we found a slurry of genes that were different. You can see CCL2, CCL7, chemokine genes, and an interferon gamma response was what we found. And then we also did the same experiment in vivo, where we're taking macrophages that were exposed to MEK1 high exosomes versus low. And now when you look at the in vivo gene induction of these macrophages, you can see lots of interferon-induced genes here, and you have a very strong interferon signature. Okay, so in vitro treatment of macrophages induce chemokines and a relatively weak interferon response, but in vivo, this would be in vivo induce a very strong interferon response. And I want to show you that again, here's in vitro. You can see the top genes here are things that recruit more immune cells. Okay. And the top genes induce in vivo. This is now the top 11 genes. 8 of them are associated with interferon. Okay. And so we think these MEK1 high exosomes are inducing an interferon signature in the tumor. Is that functional? We then treated the tumor with anti-interferon. And so you have an MEK1 low tumor here. This is just a progressor. You can see anti-interferon doesn't really do anything. But this MEK1 high tumor, it grows more slowly because it has MEK1. When you treat with anti-interferon, you can see that the tumor grows more quickly. Okay. And so our model is that we have tumor cells, and if the tumor cells are progressors, they are making these exosomes that can't transfer MEK1 into macrophages, and these macrophages end up being pro-tumor. But if you have a regressor type tumor that can make MEK1 exosomes, or you can force a progressor to put MEK1 into its exosomes, you can transfer MEK1 into the macrophages. This will induce chemokines that recruit NK cells and T cells that make interferon, and this can induce an anti-tumor immune response. So where do we want to go with this? So when you look in the literature, you can see that MEK1 is known to actually modulate macrophage activity. Our model is that we're actually potentially changing MEK1 activity in the macrophage by giving it exosomes with MEK1. That's kind of a novel paradigm, that exosomes can actually bring signaling molecules into other cells and induce their activity. We haven't proven it, but that's certainly what we'd like to do. But if you look in the literature, MEK1 has activity in macrophages. And if you inhibit MEK1, for example, you can promote an M2-like property, which this paper at least agrees with our idea that MEK1 would promote more like an M1-like activity. And so you inhibit it, you promote these wound healing properties of macrophages. And we'd obviously like to engineer liposomes to deliver MEK1. We want to look at human tumors. We actually did study human tumor exosomes. And in fact, we were never able to find MEK1 in human tumor exosomes. And maybe this is because patients all have progressor tumors, right? And if they had small regressor clones, maybe they don't make enough exosomes for us to detect them. This also suggests that if we were to make a better MEK1 inhibitor, we should target it to tumor cells. Because if we give a systemic MEK1 inhibitor, it could inhibit macrophage activation. And so targeting MEK1 inhibition to tumor cells might be a more specific and more efficacious way to perform immune therapy. Okay. And of course, we'd like to also study other differences between regressor and progressor exosomes given our model system. Okay, so I do have a little bit of time. I'm going to move a little bit beyond cancer and talk about this, this finding that we recently published, looking at IL-17D outside of cancer. I want to pause, see if there's any burning questions. I'll also take questions at the end, probably just in 5 minutes or so.
Jack, you can go ahead.
Okay. So so I bring up this slide here showing that the role of IL-17D is in tumor immunity, antiviral immunity, and antibacterial immunity. And we've published several studies to support this. The tumor immunity study I just went over, we've challenged IL-17D knockout animals with various viruses. And in all cases, you can see that there's slightly larger lesion with vaccinia, increased weight loss with LCMV, increased weight loss with MCMV. And in group A strep, we also had decreased survival if you knock out 17D. The effects are very slight, suggesting that there's probably redundancy. Okay. But nevertheless, 17D seems to be to play a role in all of these infections. And so the question that Alan asked was, how is it induced during bacterial infection? We know that NRF2 controls it in tumor cells, and that TBHQ and other agonists can activate NRF2 and IL-17D in tumor cells. But what happens in a bacterial infection that could lead to 17D production? And so stepping back, this is the stranger and the danger model for how bacteria are recognized. They could have flagellum or other components that are recognized by toll-like receptors or other receptors that can then activate the immune system. Shown here is the dendritic cell recognizing flagellum. You could cause damage, and you can release danger molecules like uric acid molecules that can also stimulate dendritic cells. But importantly, we think that both immune cells, which are non-infected, as well as the infected epithelial cell, might actually detect infection. We know that epithelial cells can have cytoplasmic toll-like receptors, cytoplasmic pattern recognition receptors, and that they can detect and produce cytokines when infected. And so what I'll show you is that the way IL-17D is induced, especially in non-immune cells, is that you need living bacteria, not dead bacteria. Okay. And this is the main finding here. If you take peritoneal exudate cells, these are macrophages, and you stimulate with group A strep, you can induce 17D. But if you stimulate with heat-killed group A strep, you can induce it as well, because a toll-like receptor doesn't detect living or dead cell, can't distinguish living or dead, right? The toll-like receptor can detect LPS or peptidoglycan and get stimulated. However, in epithelial cells, you only produce 17D when there's active infection. When you use heat-killed cells, you don't stimulate 17D production. And so this has nothing to do with burden. We've done a lot of controls here, and we've also looked beyond epithelial cells. We looked at sarcomas. Heat-killed never stimulates it. You can dilute out the live cells, and you can still get good stimulation. You can add a lot more heat-killed, and you still fail to induce 17D in epithelial cell lines. And then this is specific for 17D, but not TNF, because heat-killed can induce TNF in an epithelial cell, and it can't induce 17D. Okay. And so we hypothesized that you needed live cells because the live cells needed to divide and produce oxidative stress, and that led to induction of 17D. And so we did the simple experiment when we actually infected with IL-17D, with group A strep, and we added these ROS scavengers, then you could inhibit 17D production. Okay, so it's kind of the short story. This is not inducing death, of course. The cells are still alive, but they just can't induce 17D. And so we're putting out this kind of different model here, where we're calling the stress anger hypothesis. We need live infection, because live infection can induce metabolic competition, stress, anger, other things that produces 17D. And so you can discriminate commensals, which all have toll-like receptor ligands, right? They have LPS, peptidoglycans, but they're not growing as well, and they don't create a burden. You can discriminate between commensals and real pathogen infection, which would result in metabolic, oxidative, or replicative stress. And so in summary, the big picture of how I think about this is that we have these intrinsic hallmarks of cancer cells. They have limitless replication, they can undergo metastasis, angiogenesis. We're recently appreciating they have subcellular hallmarks, oxidative stress, DNA damage, proteotoxic stress. These all characterize cancer cells. But we also know that cancers are almost always inflamed, that they have immune system cells in there. And so what I've been able to show is that I've connected a hallmark of cancer, of oxidative stress, to another hallmark, an intrinsic to an extrinsic hallmark, through NRF2 and IL-17D. And we now know that there are other pathways that can do that. ARID1A, proteotoxic stress can also induce certain cytokines and chemokines. And so we do think that these hallmarks of cancer almost induce an immune surveillance that could lead to cancer immunoediting. Okay, and we think that 17D is one of these pathways, and the cancer, when it's edited, learns to take out 17D, and progressors learn to prevent this pathway from being activated. As far as pathogen infection, this also, I think, is a different way to think about recognition of pathogen infection. Rather than directly recognizing pathogen molecules, we're recognizing the presence of pathogen stress through oxidative or other types of stress. And that can parallel how tumor cells are recognized. And in fact, we know the immune system evolved to recognize pathogen infection, not tumor cells. And so we think that potentially many of these pathways also are operative during pathogen infection. Okay. And so with that, go ahead and thank the people who did the experiments. Tim, Robert, and Ruth worked on IL-17D and NRF2 in cancer. Alan worked on it in the context of bacterial infection. In the Exosome project, Steve, Preston, Jared, Calvin, and Christine. I collaborate with David Leiden's lab for the Exosome project, and I always acknowledge Bob Schreiber, who really got me into cancer tumor surveillance, cancer immunoediting, and continues to be a big support for all of the projects that I work on, and of course the funding. So thanks so much, and I'll take some questions.